And now to the business of the day. I call to the podium our chairman of the day, Dimitrios Salampassis, to take over the meeting. Thank you, Dimitrios.
Thank you all so much for joining us today. I'd like to warmly welcome Dr. Christos Papadimitriou. He is the Managing Director and CEO of Tessara Therapeutics. He's leading an innovative approach to drug development by using microtissue technology to mimic the human brain. Christos and his team aim at providing a more accurate method of testing for neurodegenerative diseases, moving beyond traditional animal trials. Tessara Therapeutics is a regenerative medicine company developing real brain technology to break therapeutic barriers in the biopharmaceutical industry. Dr. Papadimitriou is an award-winning qualified neuroscientist with solid experience in commercialization. He has been working in the field of regenerative medicine at world-class institutes, including Johns Hopkins Medical School, ETH Zurich, EMBL Heidelberg, and the German Center for Neurodegenerative Diseases to develop regenerative medicine applications with a commercial focus. He's an honorary research fellow at the Florey Institute of Neuroscience and Mental Health, and he holds a doctorate in neuroscience and Alzheimer's disease by the Technische Universität Dresden. Dr. Papadimitriou, thank you so much for joining us, and the floor is yours.
Good afternoon, everyone, Rotarians and guests. I'm Christos Papadimitriou, CEO of Tessara Therapeutics. I would like to begin by expressing my sincere gratitude for the opportunity to address your club today. I especially acknowledge Dr. Dimitrios Salampassis for recommending me, Mr. Stephen Oranto for extending the formal invitation, and Ms. Jo Mavros for organizing everything behind the scenes. Thank you all for your warm welcome, and it is an honor to be here with you today. Tessara Therapeutics is a biotechnology company, and in 2020, we started working on the technology to enable and accelerate the development of effective therapies for millions of patients with neurological diseases. To orient our discussion today, I will first outline the roadmap for the presentation. We'll begin with a concise overview of how medicines are currently discovered, tested, and approved. Next, we will examine new tools built from human cells or advanced computing technologies that are reshaping traditional approaches. We will then focus on brain disorders, where the gap between patient need and effective therapies is the greatest. Finally, I will show how these new approaches, particularly Tessara's platform, can open a faster, safer route to treatments. Let us start by thanking modern science and the countless professionals who have unlocked the potential for longer, healthier lives. Over-the-counter medication for cold and flu or advanced prescriptions for cancer, the progress is undeniable. These advancements owes much to regulatory agencies like the TGA Good Administration here in Australia or the equivalent FDA in the US. Such regulatory frameworks are necessary to ensure that medicines are safe and effective for human use and avoid tragedies like the sulfanilamide elixir incident that resulted in over 100 deaths in the US and many more in Europe around 1937. Since then, animal models have served as the gold standard for safety evaluation in various areas of drug development. Here is a very high-level overview of the life cycle of a new medicine. In the discovery phase, scientists identify a biological target that drives a disease, and scientists design molecules that activate or deactivate this target. In preclinical studies, those molecules are in animals to validate that have the right effect on the target and to confirm that they are non-toxic and to set an initial safe dose for human trials. During the human trials, the clinical development, the lead candidate that has gone through the filter is given to a small group of healthy volunteers to check its safety. In later phases, phases 2 and 3, the experimental drug is given to larger patient groups to answer if the drug works and if it is still safe. After all these tests, regulators scrutinize all the data and will only approve a new drug if it is clearly outperforms the current standard of care. From start to finish, this journey can take from 10 to 70 years and can cost up to $2 billion US. And only around 95% of candidates never reach the patients. And more than 70% of those failures start or stem from the weak science in the early stages of the preclinical phase. So selecting the wrong candidate at the beginning of this long journey can waste billions of dollars later. At the moment, we experience a very strong move towards improving the drug development process that we just saw before. In December 2020, President Biden signed the FDA Modernization Act and ends the blanket rule that every investigational drug must first to be tested in animals before the trials. Developers can now submit data from alternative non-animal human methods. On April 10 this year, the FDA went a step further. It announced a program to phase out animal tests for monoclonal antibodies, which is a type of therapies commonly used for neurological diseases. So let's review some examples of human-relevant non-animal models, or short. Microfluidic devices that include cell culture compartments are called microphysiological systems and have given rise to organ-on-a-chip technologies. These devices model various human organs on one chip and their interactions as well. And a few years ago, the FDA approved the start of a clinical trial based only on data derived from such a microphysiological system. Another pillar is the in silico drug development, supported by advancements. Better now? Supported by advancements in machine learning and artificial intelligence, these computational models can predict drug reactions and effects bypassing traditional testing. Recently, a drug that discovered and designed with artificial intelligence entered clinical trials. One of the most exciting developments in this is organotypic cultures. These are advanced cell culture techniques that clearly replicate human organ structures and technologies have been part of the drug development landscape for years, but only recently advancements have significantly enhanced their accuracy and reliability. You see here at me, there is a beating cardiac organoid that is used to expedite and de-risk drug development. A recent report from CSIRO Futures suggests that our nation stands on the cusp of a significant opportunity. Apart from the medical advancements that these new technologies are bringing, we have the potential to harness approximately 1.5 billion in growth by accelerating the adoption of non-animal models. As I shared with you previously, we are living longer lives, yet many of us are not living healthier ones. Recent data show that neurological conditions have overtaken heart disease and cancer as the leading cause of ill health and disability worldwide, underscoring an urgent need for better ways to study, prevent, and treat diseases for the brain and the nervous system. So within that enormous burden of neurological diseases, dementia, especially Alzheimer's, is among the largest drivers of poor health and disability worldwide. Dementia is not just one disease. It is an umbrella term for a cluster of symptoms that include memory loss, impaired reasoning, behavioral change, and many others. Several distinct diseases can cause those symptoms. The main ones are vascular dementia, Lewy body dementia, frontotemporal dementia, and most importantly, Alzheimer's disease. Alzheimer's alone accounts for roughly 60 to 80% of all dementia cases. And despite its significance, we only know the cause of Alzheimer's for about 3% of all patients that have rare inherited familial cases of Alzheimer's disease that appear early in life. For the remaining 90+%, the so-called sporadic Alzheimer's, there is no single clear cause. Instead, it arises from a mix of genetic risks, environmental exposure, and lifestyle factors. So let me walk you through what actually goes wrong in an Alzheimer's brain and why it is so hard to model this disease in the lab. First, we have a small protein fragment that is called amyloid beta 42. And this small protein piles up in the brain because the brain either makes too much of it or it clears it too slowly. As a result, this peptide sticks together and forms hard plaques inside the brain around neurons and comes also inside neurons at some point. These plaques are toxic and block the communication between neurons. And the stress and toxicity from these plaques changes, as a result, another protein that is called tau. Tau normally supports the structure of the neurons, give neurons a shape, and helps them function properly. And in this setting, it twists into tangles inside the neurons, forming the so-called neurofibrillary tangles. So plaques and tangles spread in the brain slowly over decades.
No, I can't
and
do
start
anything
killing
about it.
neurons. And
They're
then
here.
we have memory loss and other symptoms we see in Alzheimer's patients. The problem is that mice and other animal
You see
models
it there.
never develop plaques or tangles on their own. So to make a model, scientists genetically insert rare familiar genes inside these animal models, which causes a quick, early onset of the disease. Only relevant in few percent of patients, not the 97% of patients that have the sporadic version of the disease. So because the signs from the models we have do not translate to what we know in humans, nearly every Alzheimer's drug that looked promising in animals has failed in clinical trials. Tesara's answer to this problem are the real brain neural microtissues. Each sphere on the left that you see there, it is a miniature human brain, cortex, that is grown from stem cells. And what you see in this video that it rotates, it's not a cartoon. These are actual human neurons that communicate and create networks, giving us a window into how treatments work in the brain. Our solution is not perfect, but helps us to get closer to finding cures for neurological diseases like Alzheimer's. To make these mini brains, we get blood or bone marrow cells from donors, and we turn them into human neural stem cells. Then we mix them with special biomaterials to form tiny spherical scaffolds. During a period of about 3 weeks, the cells that we mix with the biomaterials start to build their own brain tissue. And then, after about 3 weeks, we can then use them for a variety of tests to help us understand if a therapeutic candidate is safe and if it works as it's supposed to. From day one, we have engineered this technology for scale. A single robot that we have mixes our biomaterials and cells and is able to produce up to 15,000 microtissues per day, which is an industry-leading reproducibility achievement. After the encapsulation and over a period of 3 weeks, the neural stem cells that we initially add into the mix follow a maturation process similar to human neural development, giving rise to multiple neuronal subtypes. On day 0, each microtissue shows a uniform cell distribution. After about a week, the first networks of neurons start to appear, and later we can see the development of neural bundles. So we normally use these microtissues after about 3 weeks of maturation, and we can maintain them for more than 6 months, allowing us to perform chronic exposure studies. When the microtissues mature, they create a variety of different brain cells, like what we have in our human brain. So here you can see some of the characterization of different markers where every color corresponds to a different type of a brain cell. This heterogeneous population of brain cells is important because some brain cells activate and some other cells inhibit brain functions. And we need to capture these connections very accurately so we can have a true network effect and understand how new treatments work in the human brain. Now on the right side, you can see how neurons light up when they are stimulated with drugs that affect neural signaling. Also, when we place these microtissues on a grid of electrodes, we pick up the bursts of electricity that neurons use to talk to each other. And you see that you see this neural burst as the spikes that you see in the video behind me. So this type of data tells us how the cells are sensing and reacting to specific drugs, just as they would in the human brain. And these are unique insights that we cannot get when we administer the drugs in animals or in humans. So Tessara's first commercially available products are the RT-Brain, a model of healthy version of the brain, and the AD-Brain, model of Alzheimer's disease. These will be followed by models of neural development, traumatic brain injury, and brain cancer. To make the microtissues that mirror the sporadic Alzheimer's pathology, we start with the same population of cells, like the healthy ones, no genetic manipulations. And you can see this addition of amyloid beta 42 that we are adding as green dots with the neurons. So throughout the process of maturation, these Alzheimer's brain tissues grow up normally and show a very patchy neural network formation compared to the healthy version. And that results in neural network loss and other symptoms that we expect from Alzheimer's disease. There are many more tests that we have performed confirming the similarity of how Alzheimer's is affecting the human brain and how it is formed in our microtissues. But for the interest of time, I will only share the most important one. This is the mother of all tests. So here in this slide, you can see a special dye that sticks to the plaques and tangles that I explained to you previously and turns them dark. On the left-hand side, you see the stained sections from Tessara's model, the AD brain. And on the right side, the same type of staining from the brain of a person who had Alzheimer's disease. And we notice the same exactly hallmarks and partners. Dark, tangled neurons with characteristic shape of candle flame and plaques scattered throughout the tissue. This is very significant because mice and other animal models do not form these tangles and plaques without genetic manipulation. Our microtissues do. And they do so when we recreate the form of the Alzheimer's disease that is most prevalent to the general population of patients. So our model captures the full cascade of Alzheimer's from amyloid accumulation to neural death in the form of neurofibrillary tangles. This is the first human-relevant, non-animal model that develops all key hallmarks of the sporadic Alzheimer's disease. Now I want to share with you some examples on how we use this platform, and sometimes I will have to get a bit technical as well. So here you can see a typical 96 well plate layout format, where in each well, we have one microtissue. And we expose the microtissues to compounds for about 7 days, or depending the experiments. And if we look closer in the images, we can observe that these compounds cause changes in the morphology of the microtissue, damage neural networks, and also they can cause tissue swelling as well. And this is a sign of inflammation. Such studies, and others not shown here, help us to select only the compounds with the best safety profile to progress into the next level of experiments. For compounds that pass the basic safety screen, we move them to detailed imaging with the microtissues. We label the exact brain cells that we care about, Then we run images through our own analysis software to measure how each drug changes the neural network morphology and characteristics inside our platform. And here we can see two close chemical relatives of the same drug. We can see the variant A and the variant B. At higher doses, both variants here increase the branching and the network effect that we see in our microtissues, while lower concentrations do not. Those measurements, together with other data, guide our to select new disease targets and the right doses for later studies. Here, you can clearly see in the microtissues with amyloid beta 42 that they are green. And also here, we expose these microtissues to the drug of the sponsor for about 7 days. Firstly, we confirm that the compounds are non-toxic in the intended concentration. So basically, the lines here that they are closer to 0 means that they are non-toxic. Then we run a series of tests where we identify two molecules that we can see here, if you can see my cursor. And these molecules have a reduction in the neurofilament light chain, which is a test used in clinical trials in patients with Alzheimer's disease. This reduction suggests an effect of the amyloid load by fluorescent imaging. And one of these molecules caused an almost 50%, here you can see 50% reduction in the amyloid load. And this data helped our customer to progress the development of this therapeutic candidate for Alzheimer's disease. So if we take this study as an example, we tested about 20 compounds to select only one therapeutic candidate. Without our technology, the sponsor would need a minimum of 60 animals, about 3 years, and millions of dollars to collect and process the same amount of data that we gathered in 4 months and at a fraction of the costs. So platforms like Tessara's allows us to select drug candidates that are more likely to provide a true therapeutic benefit and in far less time. The economics are equally compelling. Transitioning from animal models to these human-relevant platforms can cut research and development costs significantly, raise the probability of clinical trial success, and extend the profitable years in a medicine's life cycle. We are still early in this journey, and technologies like the real brain are very young, and no medicine has yet traveled all the journey from discovery to regulatory approval and market launch. However, the first candidates are advancing rapidly, and I encourage you to stay tuned for some very exciting developments. With the scientific and commercial incentives both, regulators and drug developers have every reason to adopt these technologies and bring effective treatments to millions of patients, getting us closer to live healthier and longer lives. So thank you for your time and attention, and I hope today you learned something new. There are so many things that I didn't have the chance to share with you today, and I really welcome any questions that you may have, and please reach out if you want to help our mission or to learn more about our technology. Thank you.
Thank you so much. Thank you so much. It was really great. Very, very eloquent. We've got time for a couple of questions, so feel free to raise your hands and a microphone will be to you shortly.
So my understanding from what you said is that over 90% of the pharmaceutical candidates fall by the wayside, and 70% of those 90% are ones that we maybe shouldn't have discarded. Is your technology at this stage really about identifying the ones that we shouldn't have discarded and doing more work on them?
Yes, absolutely. And maybe, I don't know if I'm still sharing my screen. So over the last 4 years, we've tested about 100 drugs, those that they have been successful in clinical trials and those that have failed. The best use of our technology is very similar to what you said, is to really find those drugs that we shouldn't have invested billions of dollars. So our technology works as a very strict filter to only discover those that have the highest chances of being successful in humans. And by testing and knowing early if it works in the human cells, then you are already de-risking a significant part of the whole journey.
Thank you so much, Dr. Papadimitriou. I commend your model and the fact that it will help clinical trials and compress time to market as well. I just wanted to know, well, would there be a working arm for neuromuscular diseases as well?
So we are not working on neuromuscular diseases because it has a very special the junction of the nerve cells and the muscles. But there are other systems that they're doing exactly that they are trying to replicate how these junctions and connection between nerves and muscles are working.
Will it extend to gene therapies? Thank you.
Yes, gene therapy, cell and gene therapy is the biggest chances that we have in many of these diseases that they are human specific. And all of these technologies that I just mentioned to you, their real cause is to really test human diseases because many of these diseases cannot be replicated or formed in animal models. So we really have a chance with these new approach methods to test if cell and gene therapy works really in humans, yeah.
Just a quick question. You mentioned there's a number of different varieties of dementia and so forth. The one with Lewy body comes with a special downside of hallucinations. Are you aware of any treatments that are being developed or worked on to deal with that aspect, the hallucination aspect of Lewy body dementia?
I'm not familiar with any treatments that they trigger the hallucinations in the brain because they can be formed from countless regions and causes that the science is not aware of. We are working with compounds that they are supposed to be hallucinogens, but they are not. But this has to do with the chemistry of the drug rather than what it happens in the brain. So the answer is no. And I personally don't know of any treatment or the cause of this symptom.
Thank you for that presentation. Just over here, I think any researcher who would be thrilled to not have to use animals if they didn't have to. And I work at the Bionics Institute, so quite familiar with the need for animals at the moment. My question is, how would you go communicating to ethics committees that your technology platform is better and as good as animal models that are needed at the moment for that early stage clinical trial?
Excellent question, and it really depends on the readouts that you're looking to have on a study. Animals will not be replaced in the next 10 to 15 years because we still need them as the last barrier for a systemic safety and toxicity test. But when it comes to specific readouts that I mentioned to you before, like amyloid plaques and tangles, these are black and white indications. These models can have the symptoms. So why to use animals in many of these experiments? It's just not justified. And our chief scientific officer is part of these committees, and now all these committees are scrutinized more all the experiments that they have to be done in animal trials in order to minimize their use and replace them in some specific extent. So it really depends on the design of your experiments and what is the outcome that you want to have from this experiment. And we see significant changes. And just on your point here, Australia, despite the small size of the scientific output and number of researchers, had the highest use of animal models globally, which is not justified. So there are significant movements that several institutes are trying to do to bring this number down.
Dr. Papadimitriou, thank you once again for your lovely presentation and for taking the time to present during this meeting. A friendly reminder that next week is our monthly evening meeting with this month, which will be held at the nearby Imperial Hotel in their treasury room. Our guest speaker will be former Peace Fellow Ike Trisberg, speaking on misinformation. It's a very topical theme, very current, and we all hope that you won't miss this one.